For maximum picture quality, it may be necessary to adjust the tracking control on your VCR.
The Calvin cycle, named after biochemist Melvin Calvin, uses carbon from carbon dioxide and energy from the light reaction to produce materials for plant growth. By exposing green algae to radioactive carbon dioxide, Calvin first detected radioactivity in a 3-carbon compound called phosphoglyceric acid. He also noticed that many kinds of plants immediately fix carbon from atmospheric carbon dioxide into 3-carbon compounds. Calvin called such plants carbon-3 plants, or C-3 plants. On paper, the Calvin cycle appears to be about 38% efficient. This means that, theoretically, 38% of the energy the Calvin cycle receives ends up in the chemical bonds of carbohydrates, with the rest of the energy lost as heat. But in reality, the Calvin cycle performs far less efficiently than expected. In fact, C-3 plants can usually convert less than 1% of the light energy they absorb into carbohydrates. To understand why this happens, we need to take a closer look at carbon dioxide. One problem is the relative scarcity of carbon dioxide in the atmosphere, only 0.03%, as compared to oxygen at 21%. Carbon dioxide must compete with the more plentiful oxygen for the attention of ribulose diphosphate. Although RUDP is supposed to combine with the carbon dioxide to produce the unstable 6-carbon intermediate, it often joins with oxygen instead. The oxidized RUDP then goes through a series of reactions which eliminates it from the Calvin cycle. So, considering all the obstacles, it's no wonder that the ability of carbon-3 plants to fix carbon into carbohydrates doesn't measure up to expected efficiency. Around the time Calvin wound up his work, evidence was mounting to suggest that sometimes carbon doesn't first make its way into 3-carbon compounds. In plants generally found in tropical and desert climates, the first organic molecule in which carbon is fixed is a 4-carbon compound. Consequently, these are known as C-4 plants. But why should C-4 plants thrive in these environments? Let's begin to answer the question by comparing the two types of plants. In both C-3 and C-4 plants, the interior of the leaf is called the mesophyll. In C-3 plants, the mesophyll cells, where photosynthesis occurs, are arranged in 2 layers. At the top is the palisade layer, and below it is the spongy layer, named for its sponge-like appearance. Embedded in the spongy layer are bundle sheath cells, which surround the network of veins spread throughout the leaf. In C-4 plants, the mesophyll cells are tightly clustered in large rings around the bundle sheath cells. These bundle sheath cells are larger and contain their own chloroplasts. However, the grana inside are few and poorly developed, and are virtually unable to carry out the light reaction. So, it is left to the chloroplasts in the mesophyll cells, with their well-developed grana, to produce the bulk of ATP and NADPH2 for the dark reaction. And to make life even more difficult, C-4 dark reactions require the involvement of both mesophyll and bundle sheath cells. Because they're so close, materials can pass from one to the other. Carbon dioxide diffuses through the membrane of the mesophyll cell and enters the dark reaction by being fixed into oxaloacetate, a 4-carbon compound. Then, NADPH2 approaches the oxaloacetate and converts it to malate, also with 4 carbons. And then, to the great surprise of the researchers who first noticed it, malate diffuses not only out of the chloroplast, but right out of the mesophyll cell and into the bundle sheath cell, where it promptly enters a chloroplast. Here, malate re-energizes NADP to form NADPH2. In so doing, the malate breaks into a 3-carbon compound called pyruvate, and to the further amazement of biologists, carbon dioxide. Now, it may seem to be a pointless exercise to pick up carbon dioxide and then release it without even using it, but there is a point. The carbon dioxide, released by the malate, then enters the Kelvin cycle. And as we know, it is the shortage of carbon dioxide that limits the efficiency of the Kelvin cycle, and consequently the production of glucose. If we look closely at the part of the dark reaction in the mesophyll cell, we see that that whole portion of the reaction is, in reality, simply a pump that ensures an abundant supply of carbon dioxide for the Kelvin cycle. There is also a second mechanism in C4 plants that improves efficiency, and this is it. After the pyruvate diffuses into the mesophyll cell, it encounters ATP and becomes phosphoenol pyruvate, or PEP. It is PEP that picks up carbon dioxide, forming oxaloacetate. Since PEP has a low affinity for oxygen, it eagerly awaits the arrival of carbon dioxide, which certainly isn't the case with the RUDP in C3 plants. As we said earlier, C4 plants thrive in tropical and desert environments, far better than C3 plants. The explanation is now within our grasp. Special pores in the leaf surface, called stomata, regulate the movement of liquids and gases in and out of the leaf. In the hot sun, these pores very nearly close to prevent excessive loss of water to the air. But this creates a tremendous problem for the plant. Now, the diffusion of carbon dioxide into the leaf is severely restricted. C4 plants, with their carbon dioxide pumps, can easily cope with the small amounts that do manage to trickle through. But C3 plants need all the carbon dioxide they can get. So choking off the supply is intolerable. Thus, in hot regions, when carbon dioxide intake is diminished, C4 plants thrive where others fail. In temperate zones, where there is little need to close the stomata, both types of plants flourish.